Hi, I'm Jill.
And I'm Ben.
And in this video, we'll walk you through the MassWateR R package, which was developed by MassBays and our partners to help water quality monitoring organizations evaluate their data for quality control, generate streamlined data visualizations, and format data for upload to the National Water Quality Portal. This tool is optimized for discrete surface water quality data for nearly 50 different physical and chemical water parameters, such as bacteria, nutrients, temperature, and many others. We'll cover how to format and input your data files, how to evaluate your data for outliers, conduct quality control review and generate a QC report, output your data to a WQX-ready format, upload to WQX, and create powerful data visualizations of graphical and map analyses on the fly.
In order to use the MassWateR, you will need to launch the RStudio application. The RStudio IDE can be downloaded from the POSIT RStudio website. You also need to download the R software from the R website. Once that is done, launch RStudio and install the MassWateR package. Packages are installed either by typing a command, like this one, in the console or using the menu path Tools, Install Packages. After you have installed the package, you don't need to do that again, unless we make an update to MassWateR and notify you. The next step in running the MassWateR functionality is to open a new script window. File, New File, R Script. This is where you will write and save the MassWateR commands. The first command should always be to start the MassWateR library. This needs to be run each time you open RStudio. To run the command, click on the line and click Control-Enter. Command-Enter on a Mac. You will see that the command will be copied to the console below. At this point, it is also a good idea to set your working directory so that it is easier to access your files. You can find the working directory address in Windows Explorer Copy and paste it to an rsetwd command. One small inconvenience is that the slash marks in the address need to be changed from forward slashes to backward slashes. Running this line sets your working directory. You will probably use the same script each time you use MassWateR, so you should save this script so that you don't need to retype. Now you are ready to use MassWateR and add more commands to your script. To use MassWateR, you will need to create 5 files containing your data and metadata. Results data, DQO accuracy, DQO frequency, site list, and WQX metadata. You should use the templates provided for each of these. The templates are available for download on our GitHub website. Most of the files can be reused each year and only need to be created once. The results file is the main data file where you should enter all of your monitoring results and QC data. Data should be entered in exactly the same format as the template with the same columns and same column order. Note that the instruction sheet has a detailed instructions about how to fill in each column. The first column is the monitoring location ID. The activity type must be entered for each row. Monitoring results would either be field measurement observation or in situ measurements or sample routine for lab measurements. You can see the pick list options for QC activity types. Enter a date and time for each record. For depth, you can either enter an exact depth or a relative depth. Enter the characteristic name, result value, and unit of measure. Be sure to use only values from the pick lists. For the result value, you can also enter BDL for below detection limit or AQL for above quantitation limit. The quantitation limit field is only used for BDL and AQL results. The QC reference value is used for QC duplicates and as a reference value for QC calibration checks and spikes. Result measure qualifier is used if you need to qualify a result as questionable. The result attribute is an optional field that can be used to group results based on a common attribute. The collection method ID is required for water samples. You must use an official WQX method ID. You will probably have one project ID for all of your sampling. Local record ID is an optional free-form field for referencing your local IDs, and result comments are optional. The DQ accuracy file is where you enter the threshold values from your QAP for each type of quality control check. All cells must either have a value following the formats in the instructions tab or the text NA. MDL and UQL are the default minimum detection level and upper quantitation limit that are used if results are below detection limit or above quantitation limit. These can be overwritten with the quantitation limit field in the results file. For some parameters, you will want to define different thresholds for different value ranges. This is what the value range column is used for. Be sure to format the DQOs as described in the instructions. The DQO frequency completeness file is where you enter target QC frequencies and overall data completeness targets. For this example, the target frequency of 10 for pH field duplicates means that you need to conduct field duplicates for at least 10% of your total number of samples. The site list file is simply a list of all of your monitoring locations with lat/lon coordinates. Location group is an optional field that can be used for grouping sites. And the WQX meta file is the final file where you enter various metadata required for WQX. Please refer to the instructions tab for details about each. When you have formatted all of your input files, you are ready to start using MassWateR in R. The first step is to run a sorting function on your results file. This will quickly show you if there are any typos or odd entries in your data. In your R script, type the read MWR results view command and run it. This outputs a CSV file with columns matching the results file, and in each column, an alphabetically sorted list of all of the unique values in that column. This will quickly show you if there are any typos or odd entries in your data. Check the ends of the lists to make sure there are no strange values. And check for typos, too. When you are comfortable with the values in the results file, you can load it into MassWateR as follows. To load the files, type a read command for each file. The read MWR results command is for the results data and assigns the results data table to an object named resdat. The read MWR ACK command is for the DQO accuracy file and is assigned to an object named ACKDAT. And so forth. When you have read all of the files, add them to a single envelope object called fsetls. This makes it easy to call all files at once in future commands. You can view the data in any of the files by typing view and the object name. The data will display in a data table viewing tab in RStudio. You can also click here to open a viewing tab. Once your data is loaded, the first thing you should do is analyze your results for outliers or for strange values. In RStudio, type the Analyze MWR Outlier command. To run this command in its most basic form, define the dataset envelope we created earlier, fsetls, select a parameter. Here we are using TP for total phosphorus. Select a graph type, such as box or box plots. Other options are jitter, which showed data as points, or jitterbox, which shows both box plots and points. And set grouping, such as month or week. Run this to view a graph of the total phosphorus data grouped by month. There are many other ways to refine this command, such as adding a date range with the DTRNG argument, or changing the label size with the lab size argument. You can see all of the options available for you for this command or any other MassWateR command by calling the help screen. Type in the console $Analyze MWR Outlier to get the help screen, which shows a list of all refinements that can be added. The easiest way to view all outliers is to run the Analyze MWR Outlier All command, which will create a Word or image file with graphs of all parameters in your results file. Define the output type, directory, and file name. You can use this file to quickly scan through all of your data looking for outliers. You should research each outlier to confirm whether it is real data or not. If there is an issue with the data, go back to your data source and correct it, and then reload your corrected results into MassWateR. After resolving all questions about outliers, the next step is to generate a QC report. You do this by running the QC MWR review command, which creates a Word document with the file name qcreport.docx. Note that another method of setting the output directory is to use getWD, which stands for Get the Working Directory. This places the file in the working directory we set earlier. The raw data equals true argument specifies that we want to output not only the summary statistics, but the full set of QC checks also. After running this command, you can open up the Word document that was created. This document is fully editable, and it is in the exact format that you can submit to DEP when you submit your data. The first section is just documentation of the data quality objectives that you defined. The QC frequencies section shows the frequency of quality control checks as a percentage of total samples. The items that are highlighted in red did not meet the frequency objective. You can see the actual percentage calculations in the detailed list below. The QC accuracy section shows the percentage of QC checks that passed the accuracy objectives. Again, you can see the calculation of the percentages below. The data completeness section shows the percentage of data records that are not qualified. Note that this involves some iteration. While reviewing the QC report or the outlier report, you may decide that it is necessary to qualify some records, in which case you would make the change in your source data and reload it to MassWateR and rerun the QC report. Also note that true completeness is a comparison of successful samples to planned samples. If some samples were completely missed or censored as bad data, they wouldn't be in your results file at all. They would need to be added manually to this table, and the completeness percentage would need to be adjusted manually. The remainder of the report is a listing of all of the QC raw data. Each QC line item is listed out for reference, starting with field duplicates, then lab duplicates, etc. There is room for notes at the bottom of each section throughout the document. When the document is complete, you can convert it to a PDF and submit it to DEP or to other recipients. There are also separate functions to run each section of the QC report individually in RStudio, if you so desire, though the word report is the simplest approach. For example, the tab MWR ACC command returns the QC accuracy report in summary, or in the percentage form. The other commands are available in our GitHub vignette for QC functions.
With your data now evaluated for quality control, the next step is to generate an output that can be uploaded to the National Water Quality Portal via the Water Quality Exchange, or WQX. The function tab MWR WQX will compile data from your input files via the fset list and generate an Excel workbook titled My WQX Output in your working directory. After running the command and locating the saved workbook, you'll see 3 tabs are populated with your data. WQX requires these 3 relational tabs to describe your project, your monitoring location, and your results. In the Projects tab, you'll need to complete the rest of this row manually with project information. For the Locations tab, you'll need to assign an appropriate monitoring location type, such as river, stream, ocean, etc. You'll see we set defaults for some of the other columns, and you can keep these or update them manually. For projects and locations, you'll only need to upload these to WQX once, and will only need to do so again if your program makes changes. For the Results tab, no manual changes are needed here. If desired, you can copy the rows from this workbook into our WQX Upload template available on the GitHub webpage linked below. This template offers an enhanced spreadsheet layout with more interactive functionality, drop-down lists and prompts, and additional tabs with supporting information. To use the template, you'll copy all the rows from each workbook tab and paste values into the template. In doing so, you can see that drop-down menus become available, as does conditional formatting to indicate required fields for certain types of records. To complete your upload to WQX, you'll need to sign into your CDX account. Select WQX and navigate to Import and Submit. There, you will upload your Projects tab, Locations tab, and Results tab individually. There are custom import configurations available to facilitate this. Look for those that include MassWateR in their name. Then you can update your organization name and select the file for upload. If your upload passes all error checks, you can confirm submission. Within 3 to 5 days, your data will be in the portal, accessible to regulators and the public, and integrated into downstream tools like EPA's How's My Waterway.
We've described the key steps in the work process for preparing and submitting data. MassWateR also provides the ability to do further analysis of your data. With your data loaded into R, you can use all of R's unlimited capabilities to create any analysis possible. But MassWateR also includes 4 types of analysis that have been standardized and simplified for easy use: seasonal analysis, time series analysis, by site analysis, and map-based analysis. We will demonstrate each here. In your script, start typing ANLZ. You will see the 4 types of analysis commands and the outlier analysis commands. To run Analyze MWR Season, enter the data set fsetls. Specify which parameter to analyze. We will do total phosphorus. Define the time grouping, month or week. Define the type of graph, box plot, jitter, jitterbox, bar, jitter bar. And specify which type of reference threshold should be mapped. Freshwater, marine, or none. And remember that there are many other options that can be included. You can see the full list in the help screen by typing analyze MWR question mark analyze MWR season. Executing this command returns a graph of your data summarized by month. The time series analysis is run in a similar way with the analyze MWR date command. For this command, the grouping refers to whether the data should be grouped by site or location group. Remember the location group field in the site's input file? Or as a single total group. The loc group argument allows you to filter the data by location group. The site analysis is run with the analyze MWR site command. The arguments are similar. For all of these commands, you can filter by date range. This command returns a graph of data summarized by site. And lastly, the analyze MWR map command produces a map of your data summarized by site. There are two arguments that can be used to manipulate the base map. Add water can be set to low, medium, high, or null, depending on the level of detail you want to show for water features. And map type can be used to add some of the off-the-shelf base maps. Note the difference between high water detail and the default medium water detail. And remember to check the help screen for other options, such as north arrow, a scale bar, color palettes, label formats, and more.
Are you ready to jump in? We have more resources available to guide and assist you. Our GitHub page acts as a detailed user guide with step-by-step instructions, code that can be copied to your RStudio session, additional training materials, customization options, and templates. Use the Quick Start for brief overview of the topics covered in this video, or use the drop-down article list to jump to a more detailed review of each topic, as well as additional resources for modifying your plots, sharing data via WQX, and how to get the most out of the package if you're not a Massachusetts user. In the Resources tab, you can download data input templates and find other training materials. For additional help, you can join the MassWateR Community of Practice web forum. This is a space where package users can ask questions of the development team, request improvements, and connect with the user community. Links to the GitHub page and Community of Practice forum are shared below. Finally, the development of this package was funded by an EPA Exchange Network grant. Thanks to our lead developer, Marcus Beck, and thanks for watching.